So this video will be the second part of our discussion of basic animal plans and development. And we'll talk about how some animals have coeloms or body cavities, some don't, and we'll talk about the development of those differences in different lineages. We'll talk about two major lineages that show some pretty different developmental patterns, the deuterostomes and protostomes. And then we will also talk about animal symmetry and how that predicts basic lifestyle characteristics and behaviors. And finally, we'll talk about some developmental genetics, kind of the fundamentals of developmental genetics. So let's start this lecture by kind of reviewing some of the topics we covered last time. One of the things we talked about was the evolution of tissues through different grades of organization. And remember, this is the phylogeny of the major lineages we're following this semester. And again, the animals are derived from a group of protozoans called the choanoflagellates. And they don't have any tissues, so that's not really relevant in that comparison. But when we do start getting into tissues, into animals, so animals evolved on this branch right here. And the first animals are the poriferates. And remember, these are the sponges, and some experts believe that they do have incipient tissues at least, and other individuals think, no, they don't really quite have true tissues, but I'm going to leave it as incipient tissues. And then if we come over here, we have our first organisms that clearly have tissues, and these are the diploblastic animals, the ctenophores and the cnidarians, and they have endoderm and ectoderm. The remaining animals, by far the vast majority of animals, however, have the triploblastic condition, which means they also have mesoderm. One of the other things we talked about was the variation that animals have in the degree of yolk that they have in their zygotes. So some zygotes have just a little bit of yolk that's fairly evenly distributed across the zygote, in which case that's the isolecithal zygote form. And in those cases, we tend to have holoblastic cleavage, where the cleavage goes completely through the organism. The opposite end of that is when you have a lot of yolk, right? And so you have a very distinct animal pole and vegetal pole, and that's typically associated with the meroblastic cleavage. So if we look across animals, we see that the ancestral state of animals is likely to have the holoblastic situation with relatively little yolk. But we do see the evolution of meroblastic cleavage occurring independently in a number of lineages. And so this is an example of convergent evolution. And this is due to different life history strategies that have converged on this same pattern, as we'll talk about later in the semester. All right, let's continue our theme of looking at the variation in animals. And again, we're going to look at this from the perspective of developmental biology. So triploblastic animals, if you look at them that have the endoderm, ectoderm, and mesoderm, some of those animals don't have a body cavity, and a body cavity is referred to as a coelom. A coelom is not the digestive tract, but it's really just a tube going completely through the organism, and it's actually connected to the outside of the organism. When we're talking about a body cavity, we're talking about something that's actually embedded within the tissues of the animal. So it's a cavity that's not connected to the outside body. So some animals just don't have that. Their mesoderm completely fills the area between the endoderm and the ectoderm. So here we have endoderm, ectoderm, and we have a complete filling of mesoderm. These animals are said to be acoelomate. So coelomate just means it's an animal that has a body cavity. If you put A in front of that, that means there is no body cavity. So a flatworm would be an example of an animal that is like this. So if we then look at animals that do have a body cavity, like this pseudocoelomate here, it differs from the acoelomate by now having this fluid-filled cavity. Now, so this is basically the persistence of the blastocoel. So we're not completely filling this area with mesoderm. The mesoderm is found in these organs and muscles, and this lining on the outside of the body. The inside of the outside lining. So it's lining the inside of the ectoderm, if you cut this worm in half. So it's got this fluid-filled cavity. But this is called a pseudocoelomate, which means a false body cavity. It's not completely lined by mesoderm. We don't have mesoderm, as you can see here, lining the endoderm. We only have the mesoderm lining the outside. That's what makes it a pseudo-coelomate condition. And a roundworm is an example of this. We'll talk about roundworms later in the semester. In some organisms, however, we have a splitting of the mesoderm so that the body cavity actually forms completely within mesoderm. This is a true coelom. Sometimes it is just referred to as coelom, but in this case it is we're going to refer to it as the eucoelomate condition. So a eucoelomate, if we just compare it to a pseudocoelomate, notice now we have lining also around the endoderm. And there's actually a connection between the linings of the endoderm and ectoderm, and this is actually what causes mesenteries that actually support the organs in animals that are eucoelomates. So an earthworm is an example of a eucoelomate. This complete lining of the body cavity is what we refer to as a peritoneum. Now, if we look at the evolution of body cavities in organisms, we see that when we just look within the triploblastic animals, this isn't really relevant to the diploblastic and the sponges, but just within the triploblastic animals, at least within the lineages that we're looking at, most of these are eucoelomate. In reality, if you took an invertebrate zoology class, we would be talking about many other lineages that would probably bring the pseudocoelomate condition to becoming more prevalent here. Also, the acoelomate condition becoming more prevalent. But certainly within the animals that we're going to cover, the vast majority of them are eucoelomates. All right, well, let's look at some of these developmental differences in triploblastic organisms and how that can produce a body cavity or not, and the different ways that we can form a body cavity in eucoelomates. This has led to two major lineages in animal groups, which we refer to as the deuterostomes and the protostomes. Each of these lineages differ in four basic developmental characteristics: the patterns of cleavage, how the cells in some cases line up on top of each other, in some cases they form more of a spiral pattern in the blastula that's eventually formed. And then also, as these cells go through cleavage, when do they stop having the potential to develop into a separate individual, or when the cells have determined to become a specific part, such that if you separate those cells, you kill the embryo? The fate of the blastopore in some of these becomes the mouth, and some of them it becomes the anus, and that's actually how the terms deuterostome and protostome come about. And then also the mode of coelom formation in the eucoelomates. This only refers to the triploblasts that are eucoelomates. So let's look at a figure that kind of summarizes these differences between protostomes and deuterostomes. So the first thing that I mentioned is the cleavage. In the protostomes, we have what is called spiral cleavage, or the vast majority of them, where once we get to the 4-cell stage and then the 8-cell stage, when we start having stacks of cells to form the eventual blastula, you see that the cells above line up in the furrows of the cells below them. That differs from radial cleavage seen in deuterostomes, where they line up in nice columns. So that's one difference between protostomes and deuterostomes. The other is what happens to cells if they become separated. Even at the 4-cell stage, if you separate one of the cells in a protostome, it completely kills the developing embryo. But you see, that's how we get identical twins in a deuterostome. You separate these cells at the 2-cell stage, you get identical twins. If you separate them at the 4-cell stage, you get identical quadruplets. And interestingly, all species of armadillos that I'm familiar with, that is how they reproduce. At the 4-cell stage, the zygote separates and produces 4 identical quadruplets. But you just can't get that in protostomes. You can't get identical twins or quadruplets because it kills the zygote. The next difference between protostomes and deuterostomes is how they actually get their name. Protostome literally means first mouth. Stome means mouth, proto means first. So the blastopore, in this case, becomes the mouth, and then as that archenteron, that primitive gastrocele, grows, the next hole that it forms would be the anus. In deuterostomes, deutero means other, so other mouth. So the blastopore here becomes an anus, then the mouth would form another area. So another mouth, another area forms the mouth, deuterostome. Now, if you just look within eucoelomates, the mesoderm forms from the lip of the blastopore, and then it spreads out and becomes split to form the coelom. That's a situation we call schizocoelous formation of the eucoelomate condition. The eucoelomate condition in deuterostomes is typically formed by outpockets of the gut forming mesoderm that then split into these bands to expand. And this is the enterocoelous condition. So make sure that you know these 4 different categories of differences between protostomes and deuterostomes. And again, so if we start off with a triploblastic animal, if we look at those that are protostomes, some of those are going to just produce acoelomates. And the mesoderm that forms from the lip of the blastopore is just going to expand to fill the entire space between the endoderm and ectoderm. In other cases, that mesoderm will expand into the blastocele, retaining a blastocele cavity, which we a pseudocoelomate condition, because it is lined on only one side, just the ectoderm side, and not the endoderm side. So that gives us the pseudocoelomate condition. But one way to form a eucoel is, again, by having these mesodermal bands that are derived from the lip of the blastopore expand and surround the entire cavity and line both the endoderm and also line the ectoderm to produce this entire peritoneal lining of this fluid-filled eucoelomate condition. And again, this is the schizocoelomate formation. This is the schizocoelomate condition of a eucoelomate, and this is typical of most protostomes. The other way, again, that you can get the eucoelomate condition is through the enterocoelous plan, where you get these kind of Mickey Mouse ears that come off of the archenteron, the embryonic gut cavity, that then split to form the same basic structure of a mesodermal lining over both the endoderm and the ectoderm, giving us this complete peritoneal line. Enterocoelous eucoelomates that you're familiar with are chordates, which include vertebrates, and also echinoderms. Most animals we're going to talk about this semester, however, are protostomes. So again, let's look at the pattern, the evolutionary pattern associated with cleavage. Remember, protostomes are, at least ancestrally, described as having spiral cleavage, and that is shown in these lineages here. But we see that there's been within the group, this is the protostomes right here, this group right here, starting from here, including all of this. If you look at that other figure I gave you at the beginning of the semester, these are the protostomes. And yeah, traditionally we've described them as having spiral cleavage, but we see that likely there's been the evolution of lots and lots of other types of cleavage, including some what are called modified spiral. But even within some of the protostomes, some have a radial-like form of cleavage. So while that generality of protostomes and deuterostomes holds, in some cases there are exceptions to it. In the case of the deuterostomes, which are over here, they all do have radial cleavage, and radial cleavage appears to be the ancestral state for animals. Again, another thing that we're talking about that distinguishes protostomes and deuterostomes is what happens to the blastopore. And remember, in protostomes, the blastopore becomes the mouth, and that's fairly consistent, at least ancestral. There have been some reversals to that where it forms the anus. In some cases, it's a completely different pattern. In some we really don't even know yet. That's what's indicated here by these really sharply hatched lines. The story in deuterostomes is actually a little more tricky. In many of them, it is associated with the anus, the deuterostome, other mouth, so the blastopore becomes the anus. But there is also quite a bit of variation in this situation, where both the anus and the mouth develop from completely different areas of the embryo. So yes, I do want you to know the basic differences between protostomes and deuterostomes, but realize that we're learning that the picture is actually a bit more complex. Let's now move on to talking about animal symmetry as animals develop, some of them have different forms of symmetry. Symmetry is associated with how you can split an animal into two mirror images. And some animals, you can do this in a variety of planes. So these are radially symmetrical animals. Their bodies can be divided into two similar halves in multiple planes, as long as you're going through the longitudinal axis. So let's say that you're looking down on a sea anemone. You can cut it down this way, left looks like right. You can cut it down this way, left looks like right, and all the way around this radius. That's why they're called radially symmetrical animals. And typically, radially symmetrical animals are sessile, meaning they're attached to the surface. They're typically aquatic, and so they're attached to a rock or the bottom of the water body that they're in. Or they might be floating, but they're not really moving in any strong directional way. At best, they have very weak swimming capabilities. Sea anemones would fit this. Jellyfish, sea urchins would fit under this category of radial symmetry. There are other animals that are basically round, so you could cut them in multiple ways and get pretty much a left and right image, but they may have just a one paired structure or a couple of structures that technically make it where you can only divide them in one plane to get mirror images. So, for example, in this comb jelly or ctenophore, it's pretty much round, right? But really, these two tentacles make it where really you can only cut it down this one plane and get one tentacle over here and one tentacle over there. So it's basically radial, but it's technically what we call, we're going to see in a minute, is called bilateral symmetry. So we kind of combine those names and we call it a biradially symmetrical animal. But as far as the kind of general behavioral characteristics of a biradially symmetrical animal, it's pretty much like a radial animal. They are either sessile, so attached to some structure, not moving at all, or if they are moving, they're floating and with very weak directional swimming capabilities. And again, the ctenophores or comb jellies are representative of this. But most animals are what we can call bilaterally symmetrical. Their body can only be divided in one plane, which we call the sagittal plane, to produce two similar halves. So this ladybird beetle here, that's the only area, this sagittal plane is the only way that you can cut it in half and get a left side and a right side that are mirror images. In fact, bilaterally symmetrical animals are the only ones that make sense to refer to a left side and a right side. Now, bilaterally symmetrical animals behave very differently from radial or biradially symmetrical animals typically. These animals oftentimes are much more active, much more characterized by movement. And if you're going to move, you should show what is called cephalization. Cephalization is the concentration of nervous systems and sensory systems into a head end. Now, why would you do that? Well, again, if you're going to be moving, you tend to move into the world head first. And that's where you can kind of see what you're getting yourself into. You can sense the environment as you're approaching it and make appropriate behavioral decisions. Cephalization is characteristic of bilaterally symmetrical animals, typically. We'll see some deviations from that, but that's a generalization that holds in large part. When you talk about bilaterally symmetrical animals, you can talk about specific relative regional anatomical terms. So if we look across the transverse plane, or what is referred to as a cross-section chopping through an animal, we can say, okay, right down the very middle of the length, the longitudinal axis of an animal, if we do a transverse plane, we can have an anterior or head end, and then a tail end or posterior end. Another plane we can talk about is the frontal plane. So the frontal plane is this horizontal one here going through this squirrel. That's basically dividing the squirrel up into a dorsal or top, or the kind of the back side, and then a ventral or belly or bottom side. You know, I use a couple of these terms here because an animal like us, so we're related to squirrels, right? We're both mammals, but we're not tetrapods. We actually stand up on our hind limbs. So you can imagine if this squirrel stood up, right? Then the frontal plane would be going not horizontally, but it'd be going vertically. So the dorsal, for us, our back is pointing backwards, and our belly side is actually facing front. Okay, so that kind of complicates things a little bit when you talk about an animal that's bipedal versus typically most animals are going to be more oriented like the squirrel, where the dorsal is going to be the top and the ventral is going to be the bottom. All right, so the next two sets of terms may be the ones that are most likely to confuse you. So medial refers to something that's kind of along the midline of a bilaterally symmetrical animal. So this spider here, this black widow, has this red stripe right along its midline. So we would say that it has a red medial stripe. Lateral means it's more on the outside of the body, away from the midline. So this skunk has two lateral lines and a medial black patch. So see, they're relative to each other. The black patch is medial compared to the lateral white stripes. But they're all referring to something on the core part of the body, whether it's closer to the center line or closer to the outside of the core part of the body. Now, two similar terms refer to the relative position of structures on appendages. So distal and proximal refer to how something is close to the trunk of the body or on the extended distant part of an appendage. So, my wrist is distal to my elbow. That's one way that you could say that. The wrist is distal. It's farther away from the body from the elbow. But we can use the same two structures and say the elbow is proximal to the wrist. The elbow is closer to the body than the wrist. Okay, so they're relative terms, and it just means which. Proximal means it's the one that's closer to the body. Distal is the term that you're comparing that is farther toward the outside of that appendage. So our fingertips are our most distal part of our arm. So let's look at the typical pattern of symmetry evolution in animals. It's really interesting. When we talk about the sponges, remember, they're just kind of like masses of cells, and they really don't even share any true symmetry. At best, they're kind of radial, but they're kind of really just blobs. The cnidarians and the ctenophores show either radial or biradial symmetry, and we'll see that they really don't move very much or move very quickly, typically. But by far, the vast majority of animals have this origin in triploblastic, bilaterally symmetrical animals, and they're much more active in moving animals compared to the sister group that includes the cnidarians and the ctenophores. But here's one of the things that we'll talk about later in the semester that's really interesting. Look at the echinoderms way up here. Why on earth did they go back to a more radially symmetrical body plan? We'll talk about how that impacts their lifestyle and their behavior. All right, so just to summarize some of the things that we've talked about, how organisms go from a diploid zygote through developmental stages, which first includes cleavage to get you this fluid-filled blastula, and then you have gastrulation to give you this archenteron or a gastrocele, and inside here we still have the blastocele that sometimes there is no mesoderm created, and we just have these two tissues, the endoderm and the ectoderm. So that's a diploblastic animal. Other cases, you have a situation where you have the additional production of mesoderm, either from the lip of the blastopore, which could be shown here, or as out pockets of the gut, as we would have in a deuterostome. But that mesoderm then can just completely fill that blastocele to make an acoelomate animal, or it could partially line just the outside. That's going to give us our pseudocoelomates, or it can completely line the blastocele from this origin at the lip of the blastopore, and that is going to give us the schizocoelous formation of a eucoelomate. But remember over here, we got the out pockets of the gut. That's the enterocoelous formation of the same basic structure with a complete peritoneal lining of a eucoelomate condition in a deuterostome. So this is, I think this is a really good summary figure that shows some of that morphological diversity that happens during development, and it gives some examples of some of the major lineages we'll be covering this semester. So I just want to finalize this lecture by talking about what actually controls the development of different body parts. So as you go through cleavage, what makes some of these cells go through gastrulation in this part versus other cells up here? Which cells become mesoderm, which cells become endoderm, ectoderm, and how do they determine which parts of the embryo are going to turn into different organs and other parts of the adult organism? Well, first of all, I want to make sure that you understand it's not due to the fact that each of these cells has different DNA or amounts of DNA. It's the same. They're all diploid, and they all have the exact same genes. That's true of all somatic or body cells in the bodies of an individual. So, what can cause that? What we're going to see is it's really just different genes being turned on and off in different rates in the different cells as they develop. So we call that differential gene expression. Gene expression is when you turn on a gene to produce a specific protein that could affect development in this case. So one of the ways that differential gene expression can occur is through cytoplasmic specification. So here we have a zygote that you can see that these different colored regions are indicative of different proteins and messenger RNAs that can kind of kickstart the formation of proteins differently in each of these parts of the zygote. So as it divides, you see the cells that are being produced have different cytoplasmic specifications. They have different proteins and messenger RNAs. So this process is called cytoplasmic specification. These different chemicals that are found there are called morphological determinants. And so the morphological determinants indicated by the yellow here will cause those cells that have become one type of tissue. The morphological determinants that are different colors are going to cause the formation of different tissues. But the cells, as they get their different morphological determinants, can communicate with each other and also induce different patterns of gene expression. And so we call that induction. Induction is how cells communicate and induce each other to express genes in different ways. So cytoplasmic specification and induction influence differential patterns of gene expression, turning different genes on and off at different rates in different cells. And as the embryo develops, we have the operation of different what are called pattern formation genes. Some of the earliest pattern formation genes help to develop just the basic orientation of the embryo. So different cytoplasmic specifications and induction will cause pattern formation genes that say, oh, make these cells the head end, the anterior end. Make these cells the posterior end. Basically just the basic orientation of the embryo, or the dorsal versus the ventral part of the body. And what we found is that there are a lot of homologous genes that work in a huge range of animals. So the genes that set the basic orientation of anterior to posterior in an insect really are the same basic genes that do so in us. They're modified somewhat, but they come from a common ancestor that had that function. In some animals, their bodies are divided up into segments. And so in this case, segmentation genes help to divide the embryo up into those individual segments. So expression of genes in different cells break the body up into segments. So we see clear segmentation in an annelid. We have some segmentation here seen in this crustacean, this lobster or crayfish. And then we even see segments in vertebrates. So the vertebrae themselves are associated with different body segments. The muscle bands in this fish are associated with different segmentation. And so there are genes that are breaking up the body, different pattern formation genes that are establishing that specific pattern. They're called segmentation genes. But really some of the most interesting research on development has looked at homeotic or Hox genes. These are genes that are active in segmented animals, and they say, okay, now that I've got this animal in segments, what is that segment going to become? Oh, that segment is going to produce a leg. Okay, well, what about these segments back here, these posterior end segments? Well, that's where we're going to start developing some of the organs associated with digestion and excretion, perhaps. So the segmentation genes break the organism up in segments, but the Hox genes, the homeotic genes, determine what each of those segments are going to develop into. And again, homeotic genes are homologous across a wide range of animals. So here we see in the mouse embryo, the colorations here are talking about different Hox genes and that are homologous or related due to a common ancestry with the same genes that are seen in insects. Notice that we have many more of these. We have multiple copies. So we're going to see later in the semester that one of the things that could explain the success of vertebrates is they went through this mutational situation where they went to polyploidy, where they doubled entire sets of chromosomes. So where we have just one set of these in a fly, we have 4 times as many of those in vertebrates. And this has allowed kind of evolution to play with multiple changes in these Hox genes to create a variety of structures in the different segments in a vertebrate. And they've done some really cool studies looking at what happens when you get a mutation in one of these Hox genes. So one of the genes is gets a mutation called antennapedia, which that mutation causes legs to grow in segments that they really shouldn't grow in. So that gene is responsible for production of a leg, but if you get a mutant of that gene in an anterior head segment, well, you're going to create, that gene is going to be expressed, and instead of getting antenna, you're going to produce legs. And so these mutations have allowed us to kind of understand where those genes are and what the function of those genes are, and that it's differential expression of those. So genes, this gene would not normally be expressed in the head end, it would just be expressed in the thorax where the legs are in an insect. So this is actually a pretty big field in zoology. It's sometimes referred to as EVO-DEVO, evolutionary developmental biology. Because if we study the evolution of developmental genes, we can understand the evolution of the morphological diversity that we see in animals. And we've known this for a long time, that there are developmental homologies that are important for distinguishing different lines of animals, and this has been studied for a number of years. So if you just look at early vertebrate development, early vertebrates look pretty darn similar. I mean, you wouldn't be able to tell really which of these was a fish versus a human, or a chick, a tortoise, a salamander. But what we see is that as we get mutations and changes in the developmental genes, that's what leads to the more specific structures later on in development associated with the production of an individual in each of these lineages. And so we've known that this has occurred for a long time before we really understood anything about like homeotic genes. But now that we do know homeotic genes, we can understand why these changes are causing these specific morphological differences between the different lineages. So again, it's a very active field with some recent advances in molecular biology have taught us a lot about evolution and the development of different body parts. So for example, a lot of the developmental genes, as I mentioned, are homologous. They in both protostomes and deuterostomes, they go way back to a common ancestor. And so it indicates that despite the fact that U-coelomate condition gets there differently in protostomes and deuterostomes, they really do share a common ancestor that likely had a U-coelomate condition. We're also finding out that where those mutations in a developmental gene may affect one trait, it may affect multiple traits. So you may have a developmental change to give you a longer leg, but it also could lead to other cascading effects like a longer appendage in another part of the body. So maybe longer legs and longer arms. And so this allows for major morphological changes to evolve in concert more rapidly. So in review, we talked about the variation in body cavities in organisms. We've got triploblastic animals that are acoelomates. We have some that have mesodermal lining just on the inside of the ectoderm, but they do have a persistent blastocele that causes a pseudocoelomate cavity. But a lot of animals we talk about are eucoelomate. And there's 2 ways of getting the eucoelomate condition, whether you're a protostome or a deuterostome, and that's the schizocoelous or the interocellous process. But in general, I do want you to know the differences between protostomes and deuterostomes and whether they have spiral or radial cleavage, whether separating the embryo kills off the embryo, the mosaic situation, or a regulative embryo in which that can produce identical twins or quadruplets, depending on where the embryo splits. And there are going to be limitations to this. Obviously, once you get to, say, the 64 cell stage, you're not going to produce that many living offspring, because that's when cytoplasmic specification and induction and pattern formation genes are already operating, right? They just happen to operate less quickly here in deuterostomes, and they have already operated here in protostomes. The other major difference that the name comes from between protostomes and deuterostomes is what is the fate of the blastopore. It becomes the anus in deuterostomes, the mouth in protostomes. And then again, make sure that you understand the difference between the schizocoelomates and the interocoelomates to get you this eucoelomate condition. Then we talked about the differences in symmetry in animals with some of the early animals, well, the first animals, the sponges being completely asymmetrical. But then the earlier lineages just after sponges likely evolved radial symmetry or biradial symmetry. But the behavioral characteristics of those as being sessile or not really swimming much. Bilaterally symmetrical animals, though, is the vast majority of animals. It's associated with the evolution of the triploblastic situation and also the behavioral characteristics that are associated with that, including the evolution of a head, which is cephalization, and a body that is more prone to movement. And different anatomical terms can be associated with bilaterally symmetrical animals, so know what anterior, posterior, dorsal, ventral, medial, lateral, and proximal and distal refer to. Then we end up talking about the genetic control of development. It's not due to different amounts of DNA content. It's due to differential gene expression. And that starts off by cytoplasmic specification and morphological determinants being located in different parts of the zygote and being partitioned into different cells during cleavage, and then cell-to-cell induction and communication. And all of these things lead to the differential expression of developmental genes. Early on, it just basically sets the pattern formation genes active to establish anterior, posterior, top, bottom, and so forth. In segmented animals, it breaks the animals into various specific segments. The segmentation genes do. And then the homeotic or Hox genes build upon that segmentation pattern by determining what is going to develop in each segment of the body. And this studying developmental biology has really told us a lot about evolution. So the next video, we're going to start our investigation of the different anatomical and physiological systems in animals, and we're going to start with the integument, or basically the skin and its associated structures.